All right, hopefully we're all looking good. First of all, I wanted to thank the Institute for having me here today to talk. I'm really excited to talk about some of the work we've been doing with some of our patient-derived cell lines using 3D bioprinting to create in vitro models of these neurodegenerative diseases. And normally this is the part where I talk about how lovely UVic is and invite everyone, but I think probably most people might be locals. But if you're not, the university's back open, and definitely you can come by into our lab if you're interested in seeing any of our bioprinters. And also there's a lot of videos now, I think, on YouTube where you can see our team in action. So probably since you're attending a community event about to learn about the different aspects of aging, I think everyone here is familiar with someone who's suffered from some of these diseases like Alzheimer's or Parkinson's and multiple sclerosis. Those of you who know me, I'm a big racing fan, and possibly the greatest race car driver of all time, Michael Schumacher, suffered a traumatic brain injury while skiing. And even with essentially his unlimited resources, he was pretty much worth hundreds of millions of dollars. And even with the best therapy that his team can buy, they still haven't really seen major advances. And so that's one of the reasons I study neural tissues, and some of the reasons we study them in vitro setting is to try and find better treatments for these different diseases and disorders, because currently, just due to the complexity of the brain and spinal cord, it's really hard to treat these diseases and disorders. And even, as other people will talk about, understanding some of the basic biology behind some of these diseases of aging as well. What our lab's really been focused on in recent years, and the work I'll be talking about today, is finding better alternatives for discovering different drugs for treating diseases like Alzheimer's and Parkinson's. So it costs billions of dollars and takes over a decade to get a pharmaceutical drug to market. Instead of reading off my slide, I will talk about the recent Alzheimer's drug that was approved after significant time and expense. It only has marginal efficacy, and after it was approved, a lot of articles came out about the politicking that were involved to actually get the drug approved. So as you can guess, when so much money and time is involved, you aren't necessarily getting the best drugs out. It's a very difficult process to actually take these drugs to market, and even if you are getting them there, they might not necessarily actually be what's best for the patients. And one of the reasons that it's so difficult to get a drug to market is that if you look at our current drug screening models, and this applies not just for neurodegenerative diseases and traumatic injuries to the nervous system, but even if you look at cancer models, a lot of these things are being done in mice and rats, and these aren't really representative of human biology, especially if you're thinking about aging, given the relative differences in lifespan between mice and rats. So a lot of these models that are used to study things like Alzheimer's might recapitulate some of the features, but they aren't really recapitulating what's going on in terms of the human biology. But given that they are sort of the best things that we've had to work with in the past, these are the current standards for getting drugs into clinic. And I will point out that we have been in talks with Health Canada, my group, and one of our collaborators who makes bioprinters across the way in Vancouver. We've actually been in talks with Health Canada about how to get some of these 3D bioprinted human tissue models into the regulatory approval pathway, so we can be testing these drugs on human cells and getting that as sort of a part into this regulatory process. And while it's not Canada, Joe Biden in the United States actually mentioned the use of these humanized tissue models, again, as being important to integrate into the regulatory pathway, because in the US, a lot of this work is being led by pharmaceutical companies that have just found it's more effective to screen in human cells, and then they do the other animal studies to get their drugs into human testing. The other thing I will point out is that in some cases you can actually get human tissues of brain and spinal cord to test drugs on, but they are in quite limited supply. And that's one of the reasons why our lab does the work it does using stem cells, human stem cells, is because this can actually create pretty much an unlimited supply of human cells that we can use for modeling and screening drugs, as opposed to having to deal with the limitations that come with using human tissue slices. Also, interestingly, one of my friends and collaborators over here in the mechanical engineering department, with COVID and with some of the restrictions, it's actually getting harder to get human tissue shipped between countries. He works on biomechanics and was trying to get some cadavers sent up here. So it's kind of interesting. So what our group is really focused on is the use of these human-induced pluripotent stem cells. A lot of people have heard of stem cells, and one of the reasons people get really excited about stem cells is that pluripotent stem cells can become any cell type found in the body, as you can see along the bottom here, including those cells that are found in the brain and spinal cord, which is what I was very interested in. I had initially trained, I'm American, and so I did my training in the US. And at the time, there's a lot of controversy around embryonic stem cells and things like that. One of the reasons when human-induced pluripotent stem cells were discovered back in 2006, one of the things that made me really excited is that we can actually have our own HIPSC lines, and that's what I'll be talking about today. So the cell lines we use when we actually bioprint our tissues come from our collaborator Håkan Nygaard's group at UBC. And so his patients who suffer from Alzheimer's, they take a blood draw, and then they actually treat those blood cells with factors, and they go back to being these stem cells that can form brain tissue, skin tissue, and pretty much any other tissue found in the body. And I actually wrote a whole book on this, which has now been 5 years ago, when I was on sabbatical in Wisconsin, because I'm so intrigued by this idea of how you can take a stem cell and actually make it into a functioning tissue that resembles the brain or spinal cord. So what our group has gotten into a lot recently, so I've always had a focus on stem cells. And then one of the ideas was, how do we make this process more reproducible and high throughput, which is a very engineer way to think about it. So about 5 years ago, a company called Aspect, who I mentioned earlier, they have this novel bioprinting technology, and they approached our group about collaborating. So we were quite excited because this provided a way for us to make our human tissue models in a rapid and reproducible fashion. So a lot of you might be familiar with, and you might have seen some of the work we did with Island Health, where we used traditional 3D printing, where you print plastic. This is my company's mascot, and axolotl. So traditional 3D printing, you have your CAD file, your design, and then you essentially extrude a plastic filament, and that makes the shape in the CAD file. The way 3D bioprinting is different is instead of using a plastic filament, we use specialized bioinks that actually keep the cells alive and functioning, similar to that they would in your body. And you print them into a shape that resembles hopefully one of the tissues that you're hoping to model. And so with our collaborators over at Aspect, we did write a perspective piece on how you can use this 3D bioprinting process to model brain tissue. And I will say that there are things like skin, and Aspect actually has a really interesting preclinical study going where they're 3D bioprinting pancreatic tissue for treating diabetes. So there are clinical applications of 3D bioprinting. And if you want, I'm happy to answer questions about that in the question answer about why I don't think we'll actually be 3D printing replacement tissue that would go into brains. Although I do think there's a very large potential for stem cell therapy to treat some of these diseases. It's just given the nature of the diseases and disorders, it's very unlikely you would actually print a structure that would be implanted. You're most likely going to be directly injecting cells into different locuses. So like with traditional 3D printing, there's a lot of things to think about when you're going to decide to 3D print a brain. There's a lot of different bioprinters out there. What cells do you want to print? And you can probably guess that we're printing neural progenitors derived from human-induced pluripotent stem cells, which in this case come from patients suffering from Alzheimer's and Parkinson's. Our lab spent a lot of time making a bioink that was really good at keeping human stem cells alive and functioning. And so that was one of the big advances that our team had made. The tissue structure is also really important. We're getting closer to printing structures that resemble the brain. I'll talk a little about Ruchi's work where we printed half domes as well. And we've also gotten some more realistic brain structures more recently that we're actually using for some traumatic brain injury studies. And then finally, you can incorporate different factors into the inks as well as the media in order to get those cells to function like they would in your brain. So the work I'll be showing today is with the Aspect system, and their 3D bioprinter is a high-end one, so it's relatively expensive. And what's really interesting about it is they have these microfluidic print heads. And so you can see here, and yes, I think it is food coloring, it has 4 different channels. So you can actually flip between different materials. And that's what you're seeing in this printed structure, where you have like a clear whitish layer surrounded by another blue layer. And what you want to use that for when you're actually engineering tissues is you can lay down different layers of cells. So for example, in your brain, there's, your neurons, oligodendrocytes and astrocytes. And so in theory, bioprinting can allow you to arrange the cells in different combinations and concentrations to mimic what the brain tissue actually resembles. And while I talk about a lot of this in the context of neural, Aspect has published a paper where they have published this trachea, which is smooth muscle, which you can see the reference at the bottom. The other really nice thing about the Aspect system is with the microfluidic print head, it's really gentle on the stem cells. So prior to our work publishing with the Aspect system, most of the bioprinting papers that have printed these stem cell-derived neural tissues saw very poor viability, with only about 50% of the cells surviving, and they weren't really behaving in the same way that your brain tissue functions, because your brain processes a lot of information using both electrical and chemical signaling, and it really needs to have like a large number of cells that are properly functioning in order to achieve those signals. Our lab, and what we've now been commercializing through my company Aspect, had developed a really novel fibrin-based bioink. So essentially when you cut yourself and a blood clot forms, that's fibrin in action. And it's obviously highly biocompatible, being blood-derived. Cells really like to grow in it, but it's very difficult to bioprint. And so one of our major advances that my lab has made that we're now commercializing was making a version of fibrin that you could actually use with all these bioprinters to print our neural stem cells. And in fact, this paper where we published our ink was selected as one of the methods of the year for the journal. So we were quite proud of it. And it was done by Emily and Lila Abelseth, who are an amazing team of sisters who both worked for me at UVic as well as at my company. So as I mentioned, my senior PhD student Ruchi used this system to print these miniature domes, which resembled half brains. And this came out in Frontiers in Bioengineering and Biotechnology back in 2020. And this paper's been cited over 40 times in the year it's been out because people were so interested. And essentially what she did was we had these neural progenitors and half domes, and then we also had these drug-releasing microspheres that essentially told the neural stem cells to differentiate and form brain tissue. And so this paper generated a lot of excitement just because it was a unique shape that hadn't been printed before, and also due to the maturity of the cells that we were able to get inside of our constructs, because we were able to culture these for 30 days and 40 days, which in the field of bioprinting, people just really hadn't been able to culture constructs that long. And again, as we use these as a tool to study diseases like Alzheimer's and Parkinson's, it's really important to be able to keep them alive for a while, because then you can actually monitor the symptoms of these diseases, at least as they're reflected in our tissues over time, and then potentially screen for drugs to reverse those symptoms. And here's just some of the staining. So essentially, she stained for tyrosine hydroxylase, which is a dopaminergic neuron marker, which is the cells that are affected during Parkinson's. And so we found that we were getting some really nice mature neural tissues when we used our special bioink with these neural stem cells. And so that's pretty much the conclusions from this, is we were able to have essentially bioprint some relatively mature miniature brains. And we are currently, Ruchi's been following up this work by printing some cells derived from Parkinson's patients to see if they also have similar levels of expression, and also characterizing some of their properties as it relates to Parkinson's. And just to further extend on that, the work, we have a second line that's really been led by Chris Lee, who just recently graduated and is now doing his MD PhD at UBC, and John Walter Shumka and Naya Kaplan in my lab, who are both biology students. So Haka Nygaard runs the Alzheimer's clinic over at UBC. And as I mentioned, his patients who have Alzheimer's, we actually get their stem cell lines, and we've been bioprinting them as well into neural tissues. So here's just showing what some of these patient-derived stem cells look like, and that we also do get, this is in 2D, so this is not in bioprinted tissues, but they do differentiate into mature cortical neurons, which is, again, one of the important cell types that we study when we're looking at Alzheimer's and Alzheimer's symptoms. Again, this was some of his 2D cultures, but he saw that when we had the cells taken from patients who had this APP mutation, they had increased tau and amyloid beta, which is what you would see in an in vivo patient with Alzheimer's. And so the fact that we had these markers we can actually track over time is exciting, because now if we can screen drugs and reverse these effects, depending on the drugs, Hakan could actually prescribe them back to his patients whose cells were bioprinting into our tissues. And so here's just some pictures of these Alzheimer's patient cells being 3D printed into similar structures that Ruchi did for creating healthy brain tissue. And you can see he cultured these for 45 days, and we actually got some really nice maturation of these tissues. So here you can see long neuronal extension, and you can see a lot of these cells are present throughout the tissues. We printed a pretty high cell density, so it does replicate very well what you would see in actual brain tissue. And I don't think I have the data here, but we found similar patterns in terms of the expression of tau and amyloid beta in our Alzheimer's lines versus control cell lines where those mutations have been corrected. So with that, as I mentioned, we've been able to, in the middle of the pandemic, we started a company because there was a lot of interest in our bioprinting work, because it really has enabled people to print these human tissue models that weren't available before. So if you're interested in learning more, or if you're just bored, our social media intern Anna is really awesome. And so we have a lot of really cool things that have been posted both on our Instagram as well as our Twitter that they've been very excited about. Our company currently has 5 employees. And we have been selling our ink to customers starting this month. So it's been really exciting. And it's been really cool because outside of neural, they've been able to bioprint a lot of different things, including bone, heart tissue, amongst other things, some fat and some other different cell types. So it's been quite good. So with that, anyone on social media, you can always follow me on Twitter if you want to learn more about what's going on in neuroscience and 3D printing. Obviously, none of this work gets done without my home base research lab, including all of my students. As I mentioned, Ruchi's my senior PhD student, and my other current graduate students include Becca and Claire, who are just really awesome. And we have a whole awesome team of interns and my lab manager, Nadia. I'd also like to thank all my funding sources, which you can see are quite diverse. And with that, I'll turn it back over to Joanne for now. And let me know if you guys have any questions. I'm looking forward to the discussion.